ago, and I took the MCAT in June. Pranava, do you want to introduce yourself?
Yeah, I graduated in May and I went to Temple University. Now I'm doing a master's in biotechnology and I'm applying this cycle. So if you guys have any questions for us, feel free to email us later about anything. And if you want to interrupt us at any time, that's fine. And if you think you hear something that's wrong in what we say, just speak up and correct us and we'll figure it out together.
Yeah, please. I want this session to be as interactive as possible, so I encourage you to raise your hand or unmute yourself. So please speak up. So before we start, please consider donating to our GoFundMe. We'll send you the link on the post-session email, but please donate $5 so that we can cover the website and advertising costs, help us reach out to more students, and continue to provide free tutoring and help. So like I said, today's session is about enzyme kinetics. The topics we'll cover today are the Michaelis-Menten plot, the Lineweaver-Burke plot, inhibition types, the Michaelis-Menten equation, terminology, and cooperativity. So please take notes along the way as we will be asking you questions and we want you to participate and also ask us questions. So don't worry about writing every single word down because I will send you guys the slides in the post-session email and we can send you the recording if you like. All right, so let's get started. So this is the infamous Michaelis-Menten plot. On the x-axis, we have substrate concentration, and on the y-axis, we have initial reaction rate, V0, which is defined as the amount of product formed per unit time just after 0 seconds. The reaction rate depends only on substrate and enzyme concentrations. So remember to keep this in mind as the MCAT will ask you questions that requires you to know this. So in this linear portion of the graph, I'm not sure if you guys can see my cursor, but just, okay. So just the linear portion of the graph, Vmax is directly proportional to substrate concentration. So if substrate concentration doubles, then the initial reaction rate will also double. Does anyone have any questions so far? Okay. So after the linear portion, Vmax will plateau. And this plateau means that all of the enzyme's active sites are occupied or it's saturated. Vmax is a property of the enzyme and it is different with each enzyme. Another property of an enzyme that this graph tells you is the Michaelis constant Km, which is the substrate concentration at which Vmax is, at which velocity V is 50% of Vmax. Km represents the enzyme's affinity for the substrate. So an enzyme with a low Km means that it has a high affinity for an enzyme. So does anyone know why this might be?
I guess to speed up the reaction.
Okay, so an enzyme with a low Km has a high affinity for a substrate because the enzyme requires a lower amount of substrate to achieve half of Vmax. So this graph shows you how to find Vmax. So you start with finding Vmax on the y-axis here. So then you go down to 50% of this value, which would be right around here. Then you go right until you hit the line and you go down until you hit the x-axis. So that value here gives you Km. And it basically is the substrate concentration at which the reaction rate is 50% of Vmax. Does that make sense now? So would anyone like to explain why an enzyme with a high Km would have a low affinity for the substrate?
I guess it would be because a high Km means that there's a higher substrate concentration required to reach Vmax or half Vmax. So that would mean that like the substrate and enzyme don't have like as good of an affinity.
Yeah, exactly. Thank you for participating. And guys, if you want to answer a question, you can just type it in the chat if you don't feel comfortable unmuting yourself. So just as long as you guys are engaged, that's all I care about. Okay, so here's our first question. Looking at the graph to the left, does glucokinase or hexokinase have a higher affinity for glucose? I'll give you 2 minutes to work on this.
Can you guys see my poll now?
Yeah, we can see it.
Okay, thank you.
The last C is actually a D.
Yeah, sorry. Just go with that. I think that might be the case for all the questions.
Okay, so does anyone want to explain how they got that answer?
Sure, I can try explaining. So I said it was D because if you look at the curve, the Km value at which it reaches the maximum is a lot lower than it would on the hexokinase curve than the Km at the half Vmax value for glucokinase.
Yeah, exactly. So just to recap, this question is a 2x2 question, which means that you have to pick the right answer with the correct reasoning. So Km is a measure of affinity, like I said. A lower Km means that an enzyme has a higher affinity for the substrate. So we can already pick D based on that information, but we should double check by looking at the graph. So like you said, hexokinase's Km is a lot less than glucokinase's Km. So D is our answer. Does anyone have any questions?
There's a question in the chat, Ashley.
Okay, let me see it. Yeah, so if I can go back. Oh. So Km stands for the Michaelis constant, and it is defined as the substrate concentration at which the initial reaction rate is 50% of Vmax. So it's just a measure of, it's basically telling you how much substrate is needed to reach half of the maximum velocity. Does that make sense? If anyone could explain it better than me, please. Okay, thank you. All right, this is just my explanation for that. Okay. So for those of you who aren't familiar with the Lineweaver-Burke plot, it is the double reciprocal graph of the Michaelis-Menten graph. All right, so what do I mean by that? So the y-axis is a reciprocal of Vmax, 1 over V. And the x-axis is a reciprocal of the substrate concentration, which is 1 over the substrate concentration. So because this is a double reciprocal graph, increasing values of y and x are in the opposite direction, approaching the origin as I noted in red. So the intercepts are also reversed. So it's important to take the reciprocal of the intercepts to get the values of Vmax and Km, and to take that extra step of multiplying by -1 to get Km, because the x-intercept is in the negative x-axis. So the reason we use this plot is because it allows us to differentiate what type of inhibition a molecule may be by looking at the y and x intercepts. Does anyone have any questions? Okay, let me see. So what I mean by double reciprocal is that you're taking the reciprocal of the y-axis and the x-axis. So that's why it's double. Okay. The axis would be given in the MCAT. Okay, so the first type of inhibition we have is competitive. Competitive inhibitors directly compete with the substrate for the enzyme's active site. So in your own words, why do you think Km would increase? Okay, that's fair. I actually did get the answer on the slide, so I'll just go ahead and explain it. But basically, Km increases because the inhibitor reduces the amount of free enzyme available at lower concentrations of the substrate. So in other words, with the number of free enzymes lowered, more substrate is needed to supply the reduced amount of free enzyme to reach Vmax over 2. So Km increases, meaning that the enzyme's affinity for the substrate decreases, but Vmax is unchanged. Yeah, I can repeat that. So what I'm saying is that the enzyme is directly competing with the substrate. So because it's competing with the substrate, there's less of the free enzyme available. And because there's less free enzyme available, more substrate is needed to supply the reduced amount of the enzyme to reach Vmax over 2. Does that make sense? Okay. So Km increasing means that the enzyme's affinity for the substrate is decreasing, which makes sense because the inhibitor is there blocking the substrate from binding to the active site. So Vmax remains unchanged. So as you can see by my drawing below here, as you increase the concentration of the substrate, there will be enough substrate to outcompete the inhibitors. So you can still achieve your maximum reaction rate. Does anyone have any questions about this?
I have a quick question. So if you are adding more, I guess, competitors, why doesn't Km change? So it'll allow Vmax to stay the same, but why doesn't Km stay the same essentially?
So Km will increase regardless of how much inhibitor you add. So Km, like I said, it measures the enzyme's affinity for the substrate. So the enzyme will not have the same affinity of the substrate if the enzyme is uninhibited. So basically what I'm saying is that with the inhibitor directly competing for the active site, the enzyme isn't going to attract the substrate that much because the inhibitor is there competing with the substrate.
Ashley, if I can put it in other words, so the Km is the amount of substrate you need to reach 1/2 Vmax. But when you have another thing competing for the same active site, you need a higher amount of that substrate to get to the 1/2 Vmax because there'll be other things at the active site. So you just need more to crowd it out to get to that 1/2 Vmax. Does that make sense?
Yeah.
Would
Could you
do.
also think of it as Km is something inherent, whereas Vmax is depending on the amount of substrate and the concentration?
Quan, do you want to answer that? What do you mean by apparent Km and Km staying the same?
So Km is actually considered an inherent value. So for a particular enzyme with a particular substrate, you have a particular Km. If you're adding on an inhibitor, such as a competitive inhibitor, your experimentally measured Km is going to change, but your inherent Km doesn't. So this experimentally measured Km is what we would call apparent Km. So if you're actually measuring the Km using product formation or changes in UV or changes in oxygen, whatever you're using, the Km that you're seeing in your experiment changes, but the actual Km stays the same because it's inherent to that system that you're looking at.
Yeah, that was a really good explanation. Thank you, Quan and Pranava, for stepping in. So, okay. So we have the Michaelis-Menten plot on the left and the Lineweaver-Burke plot on the right. So for competitive inhibition, as I said, Vmax doesn't change. So the enzyme with the inhibitor will have the same y-intercept as an enzyme with no inhibitor present on the Lineweaver-Burke plot. On the Michaelis-Menten plot, we can see that no inhibitor in red and competitive inhibitor in blue reach the same Vmax, except Km is larger in the enzyme with the competitive inhibitor present. So we can also see that in the Lineweaver-Burke plot. So because plus competitive inhibitor, the red line, has a higher Km, the x-intercept will be closer to the origin. Does anyone have any questions about this? Okay, I have a question for you guys here. Okay, so the correct answer was A. Does anyone want to explain how they got their answer?
Yeah, I was basically looking at how you said that the y-intercept wouldn't change because yeah, Vmax stayed the same, and then the x-intercept changed because of the change in Km.
Yeah, that's great. All right, our second type of inhibition is non-competitive inhibition. So this type of inhibitor binds to the allosteric site on the free enzyme or on the enzyme substrate complex. So the inhibitor has an equal affinity for the free enzyme and the enzyme substrate complex. Oh, sorry. For an enzyme with a non-competitive inhibitor, Vmax changes, but Km doesn't. So Km remains unchanged because the affinities of the inhibitor for the enzyme substrate complex and the enzyme alone are equal. So there's a 50-50 chance that the substrate or the inhibitor will bind. And Vmax is lower because the substrate cannot outcompete the inhibitor if the inhibitor is binding allosterically. Do you want me to go back to the previous slide? Okay. Okay, so quickly going over what Vmax is, like I said, it's the maximum reaction rate of an enzyme, and it's a property of an enzyme that depends on substrate and enzyme concentration. So each enzyme will have their own Vmax. So looking at the yeah, I guess we'll start with the Lineweaver-Burk plot. So like I said, Vmax decreases, meaning that the y-intercept will be higher on the y-axis on the Lineweaver-Burk plot. So Km is not changed. So the x-intercept will be the same in both the purple and green lines on the plot. And switching over to the Michaelis-Menten plot, the enzyme and the non-competitive inhibitor in blue achieves a lower Vmax, as we said, but it has the same Km as an enzyme without an inhibitor present. Does everyone understand how we make this, how we transfer this information to the Lineweaver-Burk plot? Okay.
Sorry to interrupt. I think it would be helpful if I'm a bit confused about transferring it from the to a Lineweaver-Burk plot, but also how do you rationalize whether the Vmax is changing or and Km is unchanging in this scenario versus like the last time? Because I tend to end up memorizing it, and like I wonder if you have to rationalize it or if it's something just you find it's better to memorize.
Yeah, so you can just, I like to think of things as a story basically. So you start out with your enzyme and let's move on to, let's see if I can, nope. All right, so you have your free enzyme and your free substrates here, right? Your allosteric site is still empty, but in a case where you have this non-competitive inhibitor present, it will bind to the active site and it will bind to the allosteric site, I'm sorry. So it binds to the allosteric site and it changes the conformation of the active site. So the substrate can no longer bind on the active site and no product can be formed. In another scenario of non-competitive inhibition, the inhibitor will bind to the substrate, to the allosteric site again, but this time the enzyme will be bound to the substrate. So basically, with the inhibitor bound and the substrates bound, the substrate will be kind of trapped in this active site. So it won't be able to form the products. Does that make sense? So Vmax will be lowered because, like I said, the inhibitor is binding to the allosteric site. And if it's binding to the allosteric site, then no matter how much substrate you add on, it will not be able to compete with the inhibitor because they're not competing for the same active site. Does that make sense? Okay, so we can translate that to the Michaelis-Menten plot here. So Vmax will be lowered in the non-competitive inhibitor, but Km will be unchanged. So with the lower Vmax, we have this increased y-intercept that's like more above on the y-axis because, again, double reciprocal plot. You have to think like opposites. But the Km is unchanged, so that means the x-intercept will be the same. So Km is unchanged. I keep going to that slide, sorry about that. But Km is unchanged because the affinity for so the affinities of the inhibitor for the enzyme substrate complex and the free enzyme alone are equal. So there's a 50-50. They have the same affinity for the free enzyme or the enzyme substrate complex. So the enzyme's affinity for the substrate is not changed. I don't know if Pranava or Kwan, if you could explain that better than I can. I know that I myself get mixed up with this a lot.
I thought that was good unless anyone has questions. Which part did you want to explain differently, Ashley?
Why Km is unchanged.
So think about what Km tells you. Km tells you about substrate binding to the enzyme. So in the case of non-competitive inhibition, you have substrate binding to the enzyme, or you have substrate binding to the EI complex. If there's no difference in substrate binding to the enzyme, then you wouldn't see a change in Km. But it'll make more sense once you start explaining the alternative, which is mixed inhibition.
Okay, so now that we've talked about the Michaelis-Menten plot and the Lineweaver-Burke plot, I hope you guys are ready for a question. So just a reminder, you can rationalize this question by paying attention to the intercepts. Okay, so does anyone want to explain how they got their answer?
So if Vmax is getting lowered because it's a reciprocal, it's gonna increase on the y-axis. So I just looked at answer choice C because the line for inhibitor is higher.
Yeah, great. And so Km is also unchanged, so both lines will have the same x-intercept. Okay, so moving on to uncompetitive inhibition. Sorry. An uncompetitive inhibitor will bind to the enzyme substrate complex allosterically, so it does not bind to the enzyme before the substrate is bound. So Vmax is decreased in this case because, like I said, the inhibitor binds allosterically and limits the amount of available enzyme substrate complexes. So therefore, less of the enzyme and product are formed. And Km in this case will decrease as well because when the inhibitor binds to the enzyme substrate complex, it's increasing the apparent affinity of the enzyme for the substrate. Because if you look at this picture here, the substrate cannot readily disassociate from the enzyme. So like I said before, it's trapped in the active site. And because the inhibitor is also there, less of the enzyme and product will be formed, which is why I drew this arrow very thinly to represent less enzyme and product being formed. Does that make sense for everyone?
I have a question.
Yeah.
Sorry, you can call me Al. I tried to change my name, but my name is Arlene. Just looking at the picture, the inhibitor is attached to the allosteric site, but by default, because of that, the enzyme substrate complex can't create the product. That's why we don't see those little tiny peas.
Yeah, basically.
Right? Okay, I'm a very visual person, so I know you're explaining it, but I'm always looking at the picture instead. Okay, so because of the inhibitor binding to the allosteric site, even though the active site is bound, it just prevents the final step, which is releasing the product. So it's still, I guess you could say it's still being blocked or disabled, like Pranava said.
Yeah,
Okay.
exactly.
Okay, sorry. I just like to, make sure.
No, of course, please.
Okay.
Please interrupt me. Okay.
Okay. No. If you didn't have the tiny little peas, I'd be like, wait, is this magic? Where is where is the I? Because wouldn't the inhibitor still be attached though to the enzyme substrate complex? Or by default, does the inhibitor and the substrate disappear?
This is just so in the case where the enzyme and product form, this is just in the case where the enzyme is not inhibited by the inhibitor.
Oh yes. I'm sorry. Okay. Got it. Thank you.
No problem. So just to repeat, Km decreases in this scenario because, like I said, the inhibitor binds to the allosteric site of the enzyme that's already bound to the substrate. So the inhibitor traps this substrate in the active site, and the substrate is not ready to let go because it's trapped in the active site. So Km will decrease, and that translates to the enzyme's affinity for the substrate is actually increasing because, like I said, the substrate is trapped in the active site. It's not ready to disassociate. Does that make sense? Okay.
That makes sense. I just wanted to ask how that compares to the non-competitive example when the inhibitor is also binding to the allosteric site. Is it
Yeah, let me just go back to that slide. Oh.
So there are two examples. So I think the one on the right-hand side, the bottom right,
Mm-hmm.
the inhibitor binds.
Yeah, so it's the similar case. And the one on the left, the inhibitor is bound to the allosteric site and it changes the active site's conformation. So the substrate can't bind in the first place.
Okay,
But
and then what
yeah, in the case on the right here, the inhibitor traps the substrate in the active site and it can't form the product.
Okay, so it's the same situation.
Yeah.
So is it
Okay.
kind of 50-50%? So it's kind of why it remains unchanged?
Mhm.
Okay.
Okay, so let's review the graphs again. So the Lineweaver-Burke plot here, we have two parallel lines. The enzyme with the inhibitor will have a higher y-intercept, meaning that the Vmax is decreased. Remember, again, double reciprocal plot. And the same thing goes with Km. So because it's lowered, the x-intercept will be farther away from the origin. So because both Vmax and Km are decreased, the slope is unchanged because the slope is equal to Km over Vmax. And moving on to the Michaelis-Menten plot, we see that Vmax is lowered in the blue uncompetitive inhibition line, and so is the Km in the uninhibited case, uncompetitive inhibition case. Does anyone have any questions about these graphs? I can give you like a few seconds to write them down in your notes. Okay, question time. Okay, I think most of you guys put in your answers. So the correct answer was B. Would anyone like to share how they got their answer?
So I would say that the Vmax has to decrease. And so when there's an inhibitor, it's shown as Vmax is decreasing. And then also the Km is decreasing as well, which is why with the inhibitor, it's, I am not sure.
Yeah, so I actually see a lot of you guys put D as your answer. So you were on the right track by saying that Vmax and Km are decreased. And translating that into this Lineweaver-Burke plot, you have to remember that the axes are the double reciprocal plots. So like as you go higher on the axis, you're actually like the value itself is getting lower and lower. So like up north means that the value is decreasing. So that's why the y-intercept is higher even though Vmax is decreased. And the same goes for the x-intercept. So the x-intercept is actually farther on the left here because it's 1 over the substrate concentration. Okay, our last type of inhibition we're going to talk about is mixed inhibition. The inhibitor sorry, let me just get rid of this. So the inhibitor will bind to either the free enzyme or the enzyme substrate complex, but it has a greater affinity for one or the other. So we have two cases. Let's look at case one, where the inhibitor has a higher affinity for the enzyme alone. So this inhibitor will actually decrease the enzyme's affinity for the substrate, similar to competitive inhibition. In case two, where the inhibitor has a higher affinity for the enzyme substrate complex, it will decrease the Km because, as we mentioned, it's increasing the apparent affinity of the enzyme for the substrate. So in both cases, Vmax is decreasing because the inhibitor is binding allosterically. So whenever you see that the inhibitor is binding allosterically, you should think that, oh, that means the substrate can't outcompete it because they're not competing for the same binding site. So that's why Vmax decreases. And so like I said before, a non-competitive inhibitor is one that has an equal affinity for the free enzyme and the enzyme substrate complex. But in this case, a mixed inhibitor has an unequal affinity for one or the other. So it could favor case one more than case two or vice versa. So that's why it's often said that a non-competitive inhibitor is a type of mixed inhibitor. Does that make sense? I know that this is kind of confusing.
So I will also add that it's pretty rare. I don't know if Ashley and Quan would agree, but on the MCAT, they rarely have you identify mixed inhibition on a Lineweaver-Burke plot or a Michaelis-Menten plot because they'll have to give you more details because it could be either case one or case two. Do you agree, Ashley?
Yeah, absolutely. So if they do mention it on the MCAT, they will definitely give you like enough details to know to like recognize it. Okay, so let's look at the graphs for case one, where the inhibitor has a higher affinity for the free enzyme alone. So we can see that the blue and red lines representing mixed inhibition and the uninhibited enzyme, they intersect in the second quadrant. So this is because the mixed inhibitor has a lower Vmax, which is why the y-intercept is higher. Remember, double reciprocal plot. And it also has a higher Km. So the x-intercept will be closer to the origin. Translating this to the Michaelis-Menten plot, we see that Vmax is lowered in the blue line and Km is increased as well. I forgot to label the inhibitor on the Michaelis-Menten plot, but like the color coding is the same. So blue is with mixed inhibition, red is uninhibited. Does everyone understand? Okay, so now we'll look at the graphs for case two. The Lineweaver-Burke plot resembles that of the uncompetitive inhibitor. So because its action is similar as the inhibitor has a higher affinity for the enzyme substrate complex. So Vmax is decreased, meaning again, it has a higher y-intercept, double reciprocal plot, and Km is decreased, so the x-intercept is farther from the origin. Okay, I have a question here for you guys. Okay, that was 2 minutes. And the reason I'm giving you guys 2 minutes for these questions is because that's how much time you should usually give yourself when answering freestanding questions. Yeah, that's all I'll say about that. So would anyone like to share how they got their answer? I just realized I could potentially call on people. I'm not sure if you guys are opposed to that.
Or does anyone at least want to say what type of inhibitor they think it is before we get into the answer choices?
Would it be competitive?
Yeah. Why do you say it's competitive?
Because the Vmax isn't changing, which here it's the same y-intercept, even though the slopes are changing, which is like the Km is increasing.
Mm-hmm. And so knowing that this inhibitor is competitive, how did you eliminate or how did you choose your answer?
So when I answered this, I clicked on the wrong one, but I realized that it was A because the maximum velocity isn't decreasing, it's staying the same, as evidenced by the y-intercept being the same for all 3 lines in the Langmuir-Burke plot.
Yeah, exactly. So B, C, and D are all true statements about the competitive inhibitor. A is saying that Vmax is decreased, which is false. Therefore, it's the correct answer for this question. So yeah, so to interpret this graph, you want to compare the inhibitor. Oh,
Go ahead,
okay.
Ashley.
Sorry.
Go ahead. It was just something that I was typing, but it's unrelated to the question. So keep
Okay.
going.
Yeah. So you want to compare the inhibitor 1 times 2 times. So the 1 times 2 times is just telling you like how much the concentration is being multiplied by. So you want to compare these 2 lines to the no inhibitor presence line. So you see that they all intersect at the same y-intercept. So they all have the same y-intercept, meaning that they all have the same Vmax. The only inhibitor type that gives you the same Vmax is competitive. Does that make sense? Okay. Okay, so this is a summary of all of the types of inhibition. This table tells you where the inhibitor binds and its effects on Vmax and Km. You guys, you don't have to write all this down again. I'm going to give you the slides in like an hour. So Pranava, you could start with your portion.
Okay. Let me just share my screen.
I'll stop sharing.
Okay. So next we'll just be talking about the Michaelis-Menten equation, and we have some terminology and then cooperativity left. But just before we get started, in case anyone has to leave like on the hour at 8:30, we'll be recording it, so don't worry about that. So The Michaelis-Menten equation is another important part of enzyme kinetics, and it's tested pretty frequently on the MCAT. So I would say it's a good equation to memorize and have a good understanding of. So because Ashley explained the Michaelis-Menten plot and the Lineweaver-Burke plot, you can see that all these terms are pretty familiar to you by now. This equation basically tells you how to get VO, which is the initial velocity when you're given Vmax, the substrate concentration at a given time, and Km. And besides that, this equation is what is taken. This is the equation that you take a double reciprocal of to get the Lineweaver-Burke plot and equation. So it's kind of the basis for the rest of the stuff we learned earlier. And just at the bottom of the slide, I put in a good video of how this equation is derived from the reaction that's above it. It's not something that you need to know, but in case you wanted to know the logic behind it and how it's derived, in case that helps you understand it better, you can take a look at that. And if any question comes up while you're looking at that, you can email us. Okay, but another important part to know about the Michaelis-Menten equation is that there are certain assumptions you make in order to derive that equation. So the first one is that you essentially have to ignore the reverse reaction when you're going from the enzyme substrate complex to the enzyme and product formation. This has more so to do with how you actually get the equation. So you don't need to know too much about it. Another way to think about it is that you're measuring the initial velocity at the beginning of the reaction. So you don't have any product formed yet. So the reverse reaction going from the enzyme to the product, from the enzyme and the product back to the enzyme substrate complex is impossible. But anyway, the bottom line is just that remember that the ES to EP reaction is irreversible. And then the second assumption is called the steady state assumption. And I would have you memorize the name of this assumption as well, because sometimes on the MCAT, they won't describe what it is. They'll just say steady state assumption. So what the steady state assumption refers to is that there's an equilibrium condition established in the above reaction such that the enzyme substrate complex concentration is approximately constant. So basically, the reaction that forms the enzyme substrate complex is in equilibrium with the two reactions that dissociates the enzyme substrate complex. Okay, and feel free to interrupt me anytime. The third assumption is that the substrate is approximately constant just because in enzyme kinetics, you have so much more substrate than the enzyme that even if you're using up substrate to make product, you're basically not making much change. And again, you can think of this as you're measuring initial velocity at the beginning of the reaction. So your substrate is approximately constant, but also that just know that there's always a ton more substrate than enzyme. So this is the basis for assuming that the substrate is approximately constant. And you need to assume this because the substrate is your dependent variable. So it needs to be constant in order for you to get reliable experimental data. And 4 and 5 are pretty straightforward. You don't need to know, there's not much of an explanation behind it. It's just that what you're measuring with this is that the initial velocity of the reaction is measured and that the enzyme only exists in two forms, which is the free form and then the enzyme bound to the substrate. Okay. So I just have a question for you here. Let me launch the poll. Okay. And this one's a bit tricky, so just do your best with it. It's an actual AMC question. Okay, so we had a pretty split result for this one. So no one picked A, so that's pretty clear that, oh, sorry, that you need the steady state assumption. You can put it in the chat or interrupt me if you need an explanation for that, but I'm just going to skip it for now for the sake of time. And B is a tricky one because it's not something that we discussed, but the problem with the solution pH not being constant is that you're adding another variable when you're measuring the initial velocity. So you want your only, so in any good experiment, you only want to be changing one thing. And in this case, that's the substrate concentration. So if you change the solution pH, you change some properties of the enzymes. That's why B is incorrect. And then C is just something that you have to remember. In the nature of enzyme kinetics, the concentration of enzyme is much, much lower than the substrate. And this is an assumption of the Michaelis-Menten equation as well. So the right answer is then D. This one is tricky for sure. The point, I think this is confusing because you see the steady state assumption and then you see equilibrium here and you might get that confused. But when you're talking about equilibrium here, it's talking about the equilibrium of the whole entire reaction. So the problem with that is you're measuring the initial velocity. So this is at the beginning of the reaction. So at that point, equilibrium shouldn't have taken place. And if it does, no matter how much you vary the substrate, the initial velocity won't change. So that's the problem with that. Does anyone have any questions? I know it's pretty confusing. I just wanted to throw this question out there because it is definitely a hard question. And it's on the rarer side of questions that you'll get questions like this on the actual MCAT, but they do happen. And you should use just like process of elimination for this. Yeah, so let me
I have a question. Sorry.
Go ahead.
Okay, D, the reaction is allowed to reach equilibrium before measurements are taken. So when we do look at the enzyme graphs, we're seeing the different, the reaction progressing. So I guess that's why D would, yes, be correct, right? Because it says allowed to reach equilibrium.
So let me try to go back. Which graph are you talking about, the Michaelis-Menten or the Lineweaver-Burke?
The Michaelis-Menten.
So this one? So this is actually not the reaction progressing. This is talking about a relationship between the substrate concentration and the reaction rate. I know that's confusing because it seems like it's like one continual thing, but this is not, the x-axis doesn't represent time. It's
I
talking
know. Okay.
Okay.
Okay. What about the other graph? Because when I look at that, I see progression of inhibitors binding to the substrate.
This is the same thing. So again, no time here. The only way time factors in is the initial velocity, because velocity is a measure of product formation over time, but that's not a dependent variable you're changing. So
Okay.
it's
All right,
the
I guess I
linear
got
Burke plot is
you.
basically an inverse of the Michaelis-Menten plot. So the axes are the same, but just flipped.
Okay,
Okay.
never mind. Sorry, I was just trying to relate the two graphs and okay.
No, thank you for asking. So now I'll try to repeat why D is right, and Ashley and Quan, if you want to interrupt me, please do. So what D is saying is that the reaction should not reach equilibrium before you take the measurements. This is because what you're trying to measure with Michaelis-Menten enzyme kinetics is the initial velocity. So because you want to get the initial velocity, that's at the beginning of the reaction. Once equilibrium does take place, what happens is that the reaction stabilizes, so that even if you vary the number of substrates, or sorry, not the number, but the concentration of substrate, it'll balance itself out, if you remember Le Chatelier's principle, so that you don't really get any change in your initial velocity readings. Does that make any more sense? Okay. Okay, so the next part of this session will just be going over terminology, because this is just something that shows up not as a direct question, but you'll see this a lot in like multiple choice answers. So just some basic things. We actually haven't seen this reaction at the top right too much today, but so just to go over it a little bit, this part of the equation shows the part when the enzyme and the substrate aren't bound yet. And so when you're going to this way, this is when the enzyme and substrate have bound, and then this part is when the product has been formed and then released by the enzyme. And these are just rate constants going in one direction and then the other direction, and same thing here. But then if you remember from the Michaelis-Menten assumptions, you don't have the reverse reaction going from the enzyme and product back to the enzyme substrate complex. So that's why this is unidirectional. So the Ka, Kd, and Km are something that can be confused pretty easily. So just to go over what these are, Ka and Kd are equilibrium constants. Rate constants, but basically what Ka and Kd are opposites of each other. So 1 over Ka equals Kd, and then Kd equals 1 over Ka. So Ka is the association constant. It shows how well does the enzyme stick or bind to the substrate. So it's a measure of its binding affinity. So a high Ka is a high binding affinity. And Kd is just the opposite of that. It says how easily does the enzyme let go or not bind the substrate. So a high Kd means a low binding affinity. So I think Kd shows up just a little bit more frequently on the MCAT. So really make sure that you know that high Kd is low binding affinity. And if you just understand the meaning of association and dissociation, that'll be pretty clear to you, because association is the like enzyme and substrate associating together. So a high association constant means that they have a high affinity for each other, and then dissociation is just the opposite of that. So the last thing I threw out there is the difference between KD and KM. So we have that we often describe Km as one measure of affinity that the enzyme has for the substrate. But to break this down a little bit more, Km, we mentioned it a couple other times, but Km is actually just the substrate constant at which 1/2 Vmax is reached. So while it's a measure of affinity, its actual literal meaning is a little bit different from KD. So KD is just a rate constant from this reaction going from enzyme substrate back to free enzyme and free substrate, whereas Km is a substrate concentration at a certain point. So while you can think of both of them as having as a measure of affinity, like high KD or high KM is low binding affinity. They are slightly different in what their actual meaning is. They're not interchangeable, like KD isn't the substrate concentration at which 1/2 Vmax is reached, but they're just similar in what they represent to us. Does that make sense? Sorry.
Yes, that makes sense.
Okay. Sorry, I was just reading the chat. Okay.
Can you explain the chat, the clarity?
Yeah, Quan, do you mind taking that?
Sure. If you go back to the previous slide.
Yeah.
The equations you have written at the bottom and the equation you have written at the top, you're using slightly different notation.
You're right.
So in both, K1 is the constant we're looking at for enzyme plus substrate going to the ES complex. On the top portion, ES going back to free enzyme and free substrate is K2. But if you look at the equations on the bottom, instead of using K2, we're using K-1. So that's just a notation difference. You'll see both notations used. Normally within one question, they'll use one notation as to not confuse you though. So if you're comparing the top and the bottom, I just wanted to make sure everybody knew that K2 on the top is equivalent to K-1 on the bottom, and K3, which is the ES complex going to enzyme and free product, is actually K2 in the bottom notation.
Yeah, sorry about that, and thank you, Quan.
K3 is important. Actually, that Km equation, if you rewrite it in terms of the same constants as the top, that would be K2 plus K3 over K1. It's using a different notation, so that's why you don't see K3 on the bottom.
Oh, that makes sense. Okay, I get why. Yeah, the K3 and then the missing K3 confused me when I was comparing both top and bottom.
Yeah, I'm sorry, I missed that when I was making these. And that was a really good explanation, but I just kind of put these here so you can visually see the distinction between KD and KM, and the fact that you can see KD is the inverse of KA and vice versa, but you don't super need to know these equations. Okay. Just a couple more enzyme terminology. So we have, and on this slide, we're mainly comparing enzyme activity and specific activity. So these terms mostly come up on like experimental passages. Like passages about actual experiments. So enzyme activity is a term that you'll see come up, but it means the same thing as enzyme velocity or enzyme rate, and it'll be represented by V. And we talked about this a whole bunch earlier, but it's just how much product is generated from substrate per unit time. Well, specific activity is a little bit different. You can kind of think of this as enzyme activity per milligram of total protein in a solution. In other words, it's the rate of product production per milligram of total protein. So to give you an example, say that you have two samples that have the same enzyme activities. They're making product at the same rate. But if sample 1 has 1 milligram of total protein, and then sample 2 has 100 milligrams of total protein, which one would have a higher specific activity? Should I repeat that? I just want to see if anyone can figure that out right now. Yeah, so you have two samples, same enzyme activity, but sample 1 is 1 milligram of total protein. Sample 2 is 100 milligram of total protein. Which one has higher specific activity? Yeah, exactly. So what you do is you take the enzyme velocity, and I'll just throw out a number, 1 millimole per minute. So if you take that 1 millimole per minute, and for sample 1, you divide it just by 1 milligram, and sample 2, you divide it by 100 milligrams, so you get 1 versus 0.01. So what this tells you is it tells you the purity of the sample. So even though sample 2 is much bigger, it has the same enzyme activity, which tells you that there's a lot of other non-enzyme proteins in that total sample. So the total protein is just basically everything that's not the enzyme. So you can kind of think of this like in a lab setting when you have a certain protein sample and you're trying to purify it just to get the particular enzyme that you're looking for. Specific activity can be a measure of how pure your sample is. So you want your specific activity to be high. Does that help? Okay, thank you. All right, moving on. Okay, so these are just two other terminologies that come up pretty frequently. You have turnover number, which is denoted by this symbol KCAT, and the CAT stands for catalytic, I think. And the formula for this, if you see this on the bottom left-hand corner, it's the Vmax divided by your concentration of enzyme. So your Vmax is the rate at which you're producing product. So if you take that and divide it by the concentration of your enzyme, so what the turnover number is representing is how fast is one enzyme, or like 1 unit of concentration of enzyme, producing per unit time. So, well, Vmax gives you the rate of product production for the whole sample. Turnover number kind of breaks it down into one enzyme, or like 1 millimole of enzyme, etc. And then catalytic efficiency is also known by a whole bunch of names, and you shouldn't memorize this or understand that these all refer to the same things because they can show up in different words. So catalytic efficiency is also called kinetic efficiency, and also known as specificity constant. So the formula for this is KCAT. So this is something that we just learned, turnover number divided by your Km. So again, to keep reviewing what Km is, this is a substrate concentration at which 1/2 Vmax is reached. So when you take both of these into account, what you're taking into account is KCAT, which is how fast can a single enzyme produce product, and Km, how well it can recruit bind substrate to make product. So what you want is a high KCAT and a low Km. So overall, a high catalytic efficiency means that the enzyme works well. So if you have a high Kcat and a low Km, this tells you that even at a low substrate concentration, your enzyme is making product at a pretty fast rate. Okay, any questions on that? And a lot of this is just about memorizing the formulas, but also just understanding what they mean. I know one of the biggest challenges I had when I was trying to memorize all this stuff was that you would remember what catalytic efficiency is, but then get thrown off when I saw the term kinetic efficiency, not understanding that they mean the same thing. So while like catalytic efficiency is probably the most commonly used term, you will see these from time to time, so just don't let that throw you off. All right, just another question. Okay, so A won out just by a little bit. So this is basically an equation question. Does anyone just want to say out loud or type it into the chat? What is the equation that you need to get the catalytic efficiency?
The Kcat over Km, which in this case would just be Vmax over Km.
Yeah, perfect. So let me advance to the next slide. So like she just said, catalytic efficiency is just, the formula is just Kcat divided by Kn. But if you remember the formula for Kcat, it's Vmax over concentration of enzyme. So your formula in this case is catalytic efficiency equals Vmax over enzyme concentration over Kn. But like she said, the question specifies that all these enzymes are at the same concentration. So you can essentially ignore that concentration of E. And then so you just take Vmax over Km. If anyone wants to speak up about why they chose D, or if they see why, or if they still have any questions, you can speak up now. I chose this question specifically because you have to do these two steps. Not only do you have to know the formula for just catalytic efficiency, once you get that, you also have to know how KCAT relates to Vmax, because they don't give you KCAT directly. So often the questions you'll see on the MCAT will be layered like this. So just be sure to kind of like write out whatever formulas come to your mind and see how you can rearrange that to get what you need based on the variables that they give you. Okay, and the last topic that we have is cooperativity. So basically, cooperativity is something that happens in enzymes that have more than one subunit. So you guys might all know that hemoglobin is one of those enzymes that has multiple subunits. So what happens is that when the substrate binds to the very first subunit, that subunit changes its shape, which changes the shape of the adjacent subunit. So that second subunit either has less or more affinity for the enzyme. So in the case of positive cooperativity, which is what this slide is referring to, it makes the first subunit binding to the substrate makes the following subunits more likely to bind. So what you see is a deviation from this regular Michaelis-Menten kinetics, and you see that it takes on a sigmoidal shape instead. This is because as you get just one substrate or subunit binding, it kind of induces all the other ones to bind really quickly, and then it tapers off in a way. So just remember that the sigmoidal curve is characteristics of cooperative enzymes. And Ashley had this party analogy. It's just, you can think of it as like, as more people arrive to a party, it becomes more lively. So as more, sorry, I'm getting confused here. Ashley, do you want to take just this part, what you meant by this?
Yeah, so basically what I meant was that, so as more people arrive, like the party is getting more fun, more lively, so more people will show up. So that's how you get like this curve in the graph. But like as you're going down, that would basically represent like people are going home, the party is getting more dead, people are getting tired, so more people will leave, and then that's how you get this like dip towards the like less substrate concentration.
Thank you, sorry about that.
No worries.
Okay. And the only other thing that we'll be covering related to cooperativity is this constant called the Hill coefficient. So it doesn't show up a ton, but I've seen it in a couple questions. So it's just something that you have to memorize. When the Hill coefficient equals 1, that means that enzyme does not participate in cooperative binding or does not have characteristics of cooperative binding. So that's the one in the yellow right here. It's just your basic Michaelis-Menten style plot. And then if their Hill coefficient is greater than 1, that has positive cooperative binding. And this is where you get that distinctive sigmoidal plot. Like right here is that shape. And then when their Hill coefficient is less than 1, that's negative cooperative binding, and this is pretty rare, so I wouldn't worry about it too much other than to just memorize that Hill coefficient less than 1 equals negative cooperative binding. So basically what this is that when that first subunit binds the enzyme, it makes the second or following subunits less likely to bind to the substrate. But again, it's a rare type of cooperativity, so don't worry too much about it. So that was the end of that. I just have this one last question, and I think we'll be good to go. So let me. Okay, so again, another tough question, especially because you have to calculate turnover rate, catalytic efficiency, and look at the other two options as well. So the correct answer is D. So to work through each option, A is asking about turnover number, which is kcat. And if you remember, that's equal to Vmax over enzyme concentration. And sorry, I should have specified that in the question, but if it doesn't tell you, you can assume that you're thinking that the question is asking you about the same concentration of both enzymes. So if you think about kcat being equal to Vmax over the enzyme concentration, and you have the same enzyme concentration, the enzyme with the higher Vmax has the higher kcat. So that would be X. So because X has a greater turnover number than enzyme Y, A is incorrect. And then B is talking about catalytic efficiency. And if you look down here, the formula for that is kcat over km. And when you divide that for X, that's 30 divided by around 8. So you won't have a calculator, but you can imagine that's something around like 3 or 4. And then for enzyme Y, that's 7.9 divided by 100. So that's 0.079. So obviously the catalytic efficiency for Y is a lot less than for X. So that's why B is incorrect. And then C is asking about the affinity. And we talked about how affinity can refer to KD, KA, or KM, but out of all those, you only have KM here. So we talked about lower KM is a higher affinity for the substrate. So X has the lower KM, so X has the higher affinity for the substrate. That's why C is incorrect. And then D is you're looking at cooperativity. And the only thing that that is relevant that's given in the chart is the Hill coefficient. So with Y has a Hill coefficient of 1, that means it has no cooperativity, whereas X has a Hill coefficient of 2.80, which is greater than 1, which means it exhibits positive cooperativity. Okay. That was the end of our presentation. Does anyone have any other questions or want to go back to any slides? Thank you so much for staying. I know this was long. We'll try to be faster in future future topic sessions. Yeah, you'll have paper. It's actually like a dry erase pad. So you will have something to work with to work out the math. And yeah, so if you look at C again, C, the option says enzyme X has a lower affinity for the substrate than enzyme Y. So you know you're looking on the chart, you should look for something that denotes enzyme affinity for the substrate. So the only thing that is relevant to affinity is the Km. So if you think about it, Km is the substrate concentration at which your enzyme reaches 1/2 Vmax. So a lower Km means that you need less substrate to reach the same 1/2 of the Vmax. So that's why the lower Km indicates a higher affinity of the enzyme for the substrate in making product. Does that help, Harley? Okay, great. Okay, that's everything. Ashley, do you want to go over this slide?
Yeah, thank you guys for coming and for staying this long. I'm really sorry for taking up so much of your time, but please donate to our GoFundMe again so that we can cover our website and advertising costs and help us reach out to more students and continue with the free tutoring. And also join our Facebook group to find some study buddies. You know, you can find someone who's taking the MCAT at the same time as you, and you can find someone who basically just a study partner. Those are very helpful. And you can email